Thank you so much for joining us today at our event, Where the Living and Artificial Meet. Welcome attendees and our panelists to our event, Where the Living and Artificial Meet. The Center for Excellence in Education is a Virginia-based nonprofit that was founded in 1983 by Joanne DeGennaro and Admiral Rickover, who was father of the nuclear navy and civilian uses of nuclear power. The Center for Excellence in Education, CEE, has a mission to nurture high school and university scholars to careers of excellence and leadership in STEM. We have 4 programs for teachers and students: the Research Science Institute, the USA Bio Olympiad, the Teacher Enrichment Program, which is the program sponsoring this event, and our new program, STEM Lyceums for high school students. My name is Laura Ashley, and I will be hosting today's event. I am a program manager at CEE. All right, I am pleased to welcome our 2 panelists today, Dr. Scott Rifkin and Heidi Norton. I'll tell you a little bit about them. Dr. Scott Rifkin is an associate professor at the University of California, San Diego. His lab studies show how stochastic, genetic, and environmental variation is filtered through development and physiology to produce phenotypic variation and how evolutionary forces shape that process. His current projects in the lab include probing the dynamics and robustness of the genetic network underlining sex determination and intestinal specification in nematodes, and investigating how natural variation affects the dynamic properties of signaling and regulatory networks in yeast. Heidi Norton is a senior software product manager at Illumina, where she works to build software solutions for accurate and efficient analysis of sequencing data. Prior to her work at Illumina, Heidi was a software developer and in R&D science at Biomeme, a company that makes portable molecular diagnostic solutions. Heidi studied material science and engineering at Stanford, where she did research applying protein engineering to molecular diagnostics, and bioengineering at the University of Pennsylvania, where she studied the 3D folding of the genome. So welcome both of our panelists, Dr. Rifkin and Heidi. I'm excited to hear them speak, and I'm sure that all of our attendees are as well. I'm going to turn it over to Heidi to present to you all. Tell us a little bit about what she does.
Great. Thank you so much. I'm so excited to have the chance to talk to you all today. So today I'm going to talk to you about how we can go from DNA molecules to insights about those molecules, and really to talk through the steps that we go through for genomic data analysis. So first, I just want to level set, make sure that we all know what we're talking about when we say genomics. So genomics, at a very, very basic definition, is the study of all of a person or an organism's genes, which make up the genome. There's obviously also non-coding regions of the genome that we care about as well. We can look at the interactions of these genes with each other and with the person's environment. So I'm going to be talking to you about human genomics today, but I think we'll get a chance to hear about some really exciting non-human applications as well. So first, I'm going to introduce an example type of problem that we might be trying to solve with genomics. And then we're going to walk through that example to see how we can query the genome through data generation and then data processing, and then see how the results of these steps can get us to a conclusion that actually matters in real life. All right, so first, so I work at Illumina, as Laura Ashley shared, and at Illumina, so we're a genomics company. We make technology that helps researchers and clinicians use genomics, and our mission is to unlock the power of the genome to improve human health. So when thinking about applications that, or areas that we can work on to improve human health through genomics, one of the top areas where we can make an impact is in rare disease and pediatrics. So I just want to walk you through some numbers to kind of talk through why this is an important problem. But in the NICU, the neonatal intensive care unit where really sick babies go after they're born, about 30% of all admissions there are attributable to birth defects or genetic conditions, and up to 40% of all deaths in the NICU are because of this. So what do we mean? What do I mean when I say a rare disease? So it's a disease that affects a small number of people, so about 1 in 2,000 people, But if we look at it in aggregate across all rare diseases, these are actually quite common. There's over 6,000 characterized rare diseases, and they impact 2 to 6% of the population. So this isn't just 1 in a million type thing. This is impacting a lot of people. And really, if we're looking at where can we make an impact with genomics, this is one of those areas because about 80% of rare diseases have a genetic component. If we look at what this actually means for the patient, the average path to getting a precise diagnosis for these patients takes about 8 different physicians, 5 to 7 years, and there's often 2 to 3 misdiagnoses, meaning that the doctors and the clinical team thought that the disease was caused by a particular thing. They maybe started treatment for that, found that it wasn't working, and had to keep looking. And so this is really an area where genomics might be able to make a big impact. So let's talk about, this is a real patient story that I'm going to share with you. Sebastiana Manuel was a newborn girl with persistent seizures, and she was dependent upon feeding tubes to survive. Her diagnostic workups, including brain imaging, yielded no results. So their typical steps of diagnosing this weren't giving any information of how they could treat this patient. So could there be a genetic cause to this disease, and how will we find the answer? So just keep this example in the back of your mind as we work through the steps of how we can query the genome and maybe answer this question. So first, let's talk about how do we actually generate data that we're then going to analyze. So I first need to introduce the concept of next generation sequencing to you. Maybe you learned this in school already. I definitely didn't learn this in high school. So maybe you kind of all are already very familiar with this, but just so that we kind of all know what we're talking about. Next generation sequencing is also known as massively parallel sequencing. And that means that it enables us to read the sequence of millions or billions of DNA molecules or DNA fragments at the same time. And the reason that the kind of motivation for doing this is, the initial human genome took about 13 years to sequence. So if we look at Sebastiana in the NICU, if it takes us 13 years to sequence her genome, we're not going to have an answer in time to help her. And so technology advancements, including next generation sequencing, have enabled us to now sequence a genome in less than 2 days, or even just a few hours, depending on the technology that you're using. So the way that we do this is we take the genome, the like 3 billion base pairs in the genome, and we snip it up into much smaller bite-sized chunks that we can sequence all in parallel. So that enables us to sequence much more quickly, but we end up with a pile of data that can be a challenge to analyze. So we're going to talk through some of that. So first, I'm going to walk you through how we generate that data and then how we analyze it. So our first step would be to isolate genomic DNA from a sample. So first, we would draw blood from the patient. We would maybe collect some blood from Sebastiana. And within that vial of blood are thousands of blood cells. I just want to point out that I'm only showing red blood cells here, but we actually care a lot about the white blood cells because that's where we will actually get the genomic DNA. And so then we go through a process where we bust the cells up, we purify out all of the different molecules that we don't care about, the proteins, the sugars, the fats, and we end up with a kind of more pure solution that includes our DNA, which is about those 3 billion base pairs in the human genome. So 3 billion times thousands, we have a lot of DNA in that tube. So the next step is that we need to prepare this DNA for sequencing. So this is where that massively parallel next generation sequencing comes in. So the first step we're going to do is actually fragment the DNA. So instead of a full chromosome, we're going to chew it up into smaller bite-sized chunks that allows us to process it more rapidly in parallel. And then we're going to do some additional steps. I'm not going to have time to walk through all the details of this, but essentially we're going to add some tags of DNA at the end, have a special sequence that we know what that sequence is, that enables us to then attach this DNA molecule onto a surface so that we can sequence it. So there's some really cool steps involved in this. I encourage you to read more about this. The first time I learned about this, I thought it was really cool. So once we have our DNA molecules, so now we see these DNA clusters with different colors. Those special tags that I talked about are actually also present on the glass surface. And so when our DNA molecules come in, they can bind to the surface of the glass. And then we have basically these clusters of DNA that are attached onto the glass surface, which then are, built out into these tiles that then go across the flow cell. And so then the process of sequencing is that we're going to amplify a copy of DNA alongside its template by flowing through different colors of different fluorescent labeled A's, T's, C's, and G's. So I know this is kind of a lot of information, but then what we ultimately end up with is a beautiful picture like this. Some blues, some yellows, some oranges, beautiful speckles. And that's kind of what we get off the sequencer. So we've generated the data. And what does this actually mean for Sebastiana? What are these speckles telling us? So that's now the challenge that we're going to try to address with data analysis. So to walk you through the steps of the data journey, we're first going to take those beautiful images and identify what sequencing base that came from. So it's actually initially a kind of an image processing problem. And then we're going to basically reconstruct a sequencing read from that. So remember I told you that we cut it up into a bunch of smaller fragments. They're around 150 to 500 base pairs long. So we're actually going to end up with a text file that has all of that sequence in it. And then we're going to say, where in the genome did this sequence come from? Is it human? Is it not human? If it's human, where within the genome did it come from? And then we're going to see if there's any differences in my sequence in the sample that I care about compared to the expectation. Finally, we're going to see if there's any differences that actually matter that could be informing, could explain why Sebastiana is having these seizures. So let's talk about this first step, step 0. Let's see if actually I'm trying to get this little video to play. So instead of it actually being an image, just a static image, it's actually kind of a video. So you can see we have these speckles that are changing over time, and that's part of the sequencing process. We're taking images at each of these steps. And we have to undergo some steps called base calling, where we look at each little speckle and say, okay, that color is yellow, which means that my sequence is a T. I might not actually have the color and sequence right. I have not memorized the colors and how they map to the DNA base pairs. But we basically can say, all right, here's the confidence that I have that this base pair is represented here. And then from there, we can reconstruct our sequencing reads. So here I'm showing you a FASTQ file. So that's kind of almost your starting point of data analysis. So that's kind of why I'm showing step 0 is getting to this FASTQ file. And here you can see that there's actually this string of text, T, G, A, A, C, C, A, G. That's actually my sequencing read. Those are the base pairs. A segment of the genome is represented in that section. And then there's some more information at the top about the quality of the data and some information about the quality of the data. All right, so we did our analysis step 0, check, we've got our FASTQ file. So now let's figure out how do we make sense of that sequencing of those FASTQ files. So in order to kind of help introduce some of the concepts related to this and kind of the challenge that we face with this type of analysis, I'm going to make an analogy with a phone book. It might be kind of a cheesy analogy, but I found that it's helpful to understand kind of what the challenges are that we're working with. So let's imagine that I give you a phone book and I ask you, are all the phone numbers in this phone book correct? Oh, but I shredded it up into a million pieces. So now can you tell me, are all the numbers in this phone book correct? And that might seem kind of artificial and arbitrary, but really this is kind of what we end up with these sequencing reads, right? I told you we cut up the genome into all these little sections, and now we have to make sense of it. We have to assemble it back into something that we can actually read and actually compare it to something else. So that's what we're going to do. In this analogy, each shred of paper represents a sequencing read that we saw in those FASTQ files, right? It's 150 to 500 base pairs of DNA. And our first step, we're asking the question, where did this paper shred come from in my reference phone book? So thank goodness I didn't just hand you this shred of paper. You actually also get a reference phone book to help you. So we're going to go through each shred of paper and we're going to see where within the phone book did it come from. Are there more than one place that it could map to in this phone book? Is there one unique place it can map to? You can imagine that this would take a long time, even not just for a human to do manually, but even for a computer to do. And so there have been a lot of advancements in computer science and really cool algorithms that have been developed to be able to do this quickly and efficiently. All right, so once I've found here, I've got this little orange line showing the spot in the reference phone book. And turns out that this is actually from Illumina's phone number in the phone book. I'm next going to ask, are there any differences in the phone number from my sample compared to the reference phone book? So is the phone number actually correct? And it turns out if I look at this, it looks like we actually have one digit that's incorrect. In my sample, I'm saying the last digit's a 7, but really it should be a 6. There's one digit that's incorrect. So let's bring this back to our data analysis. So we've done the mapping and aligning and variant calling with a phone book. Now let's actually walk through how we would do that with a genome, now that you kind of understand the type of problem that we're dealing with here. So really the question we're asking is, are there any meaningful mutations in this child's genome? So the first step is to map each sequencing read to a reference genome. Instead of a reference phone book, we have a reference genome. And so we have our FASTQ file, which has our sequencing reads. And then we have a reference genome, which is from another human that's been sampled or a combination of different humans that says, here's the expected sequence that you would get for these base pairs of chromosome 1, chromosome 2, chromosome 3, etc. So that's kind of our expectation. So we have our kind of experimental condition and then our expectation. And so we take each of the sequencing reads, we align it to the reference genome, and then we end up with this other text file called a sequence alignment map, which shows us where each of those sequencing reads map to in coordinates of the reference genome. So we, in a very simple picture, we have our sequencing read, we have our reference genome, and we're kind of matching up those blue sequences and then figuring out where the coordinates are of where they line up. Okay, the next step is to identify any variants. So the question I'm trying to ask here is, are there any differences in the sequence of my sample compared to the reference? So here, it's not just finding, where in the genome this came from, it's, does my sequence have any differences? So here you can see there's a variant that we're showing in red where the expectation is a C and instead we have a T. So what does that actually look like in terms of the files that we're working with? We have our alignment file here. And again, we are going to use our reference genome. We need to compare that to our expectation. And what we end up with is a variant call file. And that kind of gives a list of every single variant that we detected compared to the reference. And there's a lot of different types of variants we care about. There's single nucleotide variants, there's copy number variants, there's regions of the genome that can be duplicated, that could be deleted. We care about all of those. So we're going to kind of make a list of all of those different variants. So we're really close to the end, but now our challenge is, how do we interpret that? So we have this text file of all these different variants. What does that actually mean for Sebastiana? So what I didn't really tell you yet about this challenge is that depending on the methods that you use, the type of sample that you have, you might end up with a list of 3 million variants that could be identified in a typical whole genome sample. So a lot of these are not going to be causing disease. If we were to compare my genome to your genome, we're going to have a lot of differences. And some of that will be because of height and hair color and, my ability, liver enzymes and all kinds of different things. So really the challenge is to sort through this list of all of these different variants that are there and hone in on the ones that might be responsible for disease. So the way that we do that is we can leverage knowledge bases to assess the impact of each variant. So as researchers and clinicians learn more and more, do more and more research and studies, they can start to add to the collective knowledge base of what we know about certain genes and how they're involved in disease, what mutations there are present in those genes, and we can actually look them up in databases that can help us wade through all this huge list of variants that we have. If there's not something that an existing knowledge base might have, or even to think about how these knowledge bases were built, you could conduct research to determine the impact of the variants of unknown significance. So typically you need a larger sample size for that. You're not just going to be looking at one patient, you're going to be looking at a group of people, but that can enable you to discover new genes that related to a certain disease or mutations that might impact that patient. So we were able, let's say that, we got Sebastiana's sample all the way through to this step of identifying any possible disease-causing variants. So let's conclude and see how this whole genome sequencing actually worked for Sebastiana. It's a success story. So rapid whole genome sequencing was able to zero in on her exact diagnosis within 4 days. And they discovered a de novo or new mutation in a gene that was known to be linked to this condition called Ohtahara syndrome, which causes seizures in babies. So they were able to change her medication immediately and control her seizures. And she was able to be discharged home on Christmas Day. Since then, she's continued to grow and delighted her parents and doctors with her progress. So this is really a success story that shows kind of the impact that studying the genome, all these years of research leading up to this that enabled us to quickly identify this variant and the impact that it has on the patient. So I think I'll end there and looking forward to the end when we might have a chance to answer some questions. Thanks for your attention.
Thank you so much, Heidi. That was really interesting. I had no idea how any of that worked. So it seems very, very tedious, but thanks to technology, I'm sure that has helped a lot with this development. So yeah, all of our attendees, if you have any questions about Heidi's presentation and about what she does, please do put that in the Q&A function. After Dr. Rifkin presents, then both of our speakers will answer any questions that you may have. All right, so at this time, I'm going to turn it over to Dr. Rifkin, who will talk more about his work.
So I'm going to tell you a story that uses some of the techniques that Dr. Norton illustrated, essentially the sequencing platform that Illumina has developed and has been a great boon for all sorts of questions in all sorts of fields of biology. So my question is, how does a new gene network originate? I'll explain what I mean by that in just a little bit. First of all, you can see the screen? Yeah, it's good. Okay, good. All right, so a central question of biology is how you get from this to this, how you get from an egg, or this to this rather. Right? And this is the predominant preoccupation of the field of developmental biology. And it's been clear for a number of years that it's a little bit too restricted, much to the chagrin of penguin enthusiasts. There's a lot of variation, both in the starting material and also in the process and the outcome. And this makes studying the development and evolution studying development, the evolution of development much richer, because we can try to find principles and mechanisms despite all this variety. So here's a bunch of different organisms with eggs and then the adult form. So we have a penguin, we have a nematode. This is a tardigrade, one of these water bears that are very charismatic. So as people have studied development, it's becoming clear that development involves several different phases. And one of the phases is to basically set up a coordinate system. So the organism sets up an organizational framework. It divides itself into spatial territories with distinct identities. And then within each of these territories, then it builds something like a gut or a leg or something like that. Over the past few decades, it's also become clear that the way that this works, or the way that this is controlled, is through what are called genetic regulatory programs. So what are these? And for genetic networks, what are these? These are groups of genes that act together. They regulate each other. They turn each other on and off. What I mean by that is, gene is a piece of DNA, but RNA is made from that, right? And you have to know, okay, which gene do you want to make RNA from at a particular place and time? And so there's this whole computation that goes on in each cell to decide which genes are you actually going to make mRNA from, and then mRNA makes protein. So which genes are going to be used in particular tissues. And then, as the organism is breaking up this itself into territories, different combinations of genes will be made in the different territories. And that's sort of that identity of that territory. So my focus for today will be this friendly neighborhood nematode, C. elegans, and its relatives. So this is about 3 millimeters long. This is a hermaphrodite with eggs. And these worms have several convenient features for both development and evolutionary biology. And I'll point out one of them in the next slide. So C. elegans, the cell lineage is highly invariant. What do I mean by that? Well, we all start with a single cell, right? And then that cell divides, and eventually it divides many times and makes an organism. Now, in C. elegans, the fate of every cell, each one of these divisions, from the starting egg to the end, each one of these is highly determined. Like, and it's invariant. It happens the same way in every individual organism. Now, this is sort of weird from our perspective because, if I would take a cell in my hand and me, and a cell in the same position in your hand and you, they would actually have come from different sources. The way that mammals do it is basically we get a bunch of cells, we cell proliferation, and then we have signals that tell cells what they're going to be. It doesn't really work that way in worms. In worms, that's just too complicated. Really, your inheritance, your mother cell and your ancestor cells, they sort of determine to a large extent what you're going to be. This is actually fairly common throughout animals. C. elegans takes it a little bit to the extreme, but it's actually fairly common in animals for this cell lineage to be very important in determining what a cell is going to be, what kind of tissue it's going to be. So I'm going to focus on this particular part of this. This is the group of cells that gives rise to the intestine. It's called the E cell, E for endoderm. Endoderm is the sort of intestinal layer. And this particular, so the intestine in C. elegans is derived completely from the descendants of this particular cell here. And these are the only ones that form the intestine. And the E cells only form intestine. So it's very, very restricted. Okay, so that's what we're going to focus on. So I'm going to just draw that bit out here. Here's a single egg. There are a bunch of divisions. We're not going to pay attention to these other divisions. We're just going to focus on these cells here. Okay, these are the cells that are going to give rise to the intestines. Okay. I'm going to show, in the next slide, I'll show you a picture of sort of what it looks like. Okay, so we have the single cell, right? It divides into two. These two cells divide some more. They have 4 cells. Here's this cell down here. It's the EMS cell. That's at a 4-cell embryo stage. That will then divide again to make an E cell and an MS cell. The E cell goes on to make the intestine. So here, here's there's 2 E cells right there. They actually go into, well, the intestine's in the middle of the organism, so they have to actually go into the middle of the organism. That's what they're doing right here. And then eventually it grows into a worm. This is all inside an eggshell right here. And then the intestine is sort of in the middle of this thing. All right. Now, I told you that a lot of things in development are controlled by genetic networks. So here's a genetic network. So what are these things? So these are all different genes. All these names here are different genes. And all these arrows mean that one gene regulates another. What that means is that this gene binds to the DNA. So that, like this SCIN1 gene would bind to the DNA in front of this gene, and it would say, okay, turn on, like make mRNA. And so all these arrows, that's the way this all works. Each gene binds, the protein binds in front of the downstream gene, and it says, okay, make mRNA at this point. Okay, so the goal here is to eventually activate this gene, L2, this gene, and then this gene activates tons and tons and tons of other genes. The proteins from that gene bind in front of lots of other genes and then make intestinal cells. So this is what this network looks like in C. elegans. And basically, by the time you have 4 endodermal cells, they all know they're going to be endoderm, and then they just build it. There's some more cell division. They basically build the worm's intestine. But what I meant by setting up territories is that, that in particular cells, in this cell and this cell, you actually have to activate these genes so that the worm knows, okay, I'm going to build my intestine here and not, where the head's supposed to be or where the muscle is supposed to be. All right, so this is the way it works in C. elegans. One interesting thing about this is that all these genes here, like the zygotic part, so these are genes that are deposited by the mother inside the egg, and then eventually the egg starts making its own activating its own genes. And these are all a particular type of gene called a GATA factor. So GATA factors are a type of transcription factor. Transcription factors are genes that regulate other genes, so they bind to the DNA and then they activate other genes. And GATA factors are a particular type of these, and they're called that because they like to bind GATA sequences. So if you look at the DNA, where the transcription factor sits down in the DNA, there should be a GATA in there. That's what they like to bind to. And what's weird is that, like humans, we have 6 of these things in our whole genome. And these worms have 6 just right here in this little, just helping it make this single gut, helping it make this single tissue. So what makes a GATA factor? So what happens is that there's actually, these are proteins. Proteins have a particular structure. We can look at the amino acids. And here's a picture of one of these GATA factors binding to DNA. So the green is the DNA here. And there's a particular part of the GATA factor that actually binds it, and it sort of wraps around and sort of has a particular structure that lets it recognize that GATA sequence. People have actually worked out what recognizes it, and it turns out there's a couple amino acids, a couple parts of the protein that are very important. We want to have these this sort of pair of C's here. There's a W sort of over there. This is just the part of the sequence of a protein. You have a pair of C's here. I hope you can see my mouse. Yeah, you have a pair of C's here with some things in between. And so this is the definition of a GATA factor. So if you have a protein that sort of that has these things, then it may very well bind GATA and may very well be able to act like a transcription factor and activate a whole bunch of genes. Okay, so what we did, we wanted to study the evolution of this network. And so what we did is we took advantage of some of the technologies that Dr. Norton mentioned. And the sort of worm community took advantage of these. And what they did is they took all these related species of these worms. So here's C. elegans here, the sort of the one that we know a lot about, but there's actually a lot of closely related species that have been identified, about 60 of them. And people sequence them. So sequence them means that they sequenced a genome from them. So we know what the genome of at least one individual from each of these different species is. Okay, and then we can compare them. So this is really nice. It's actually somewhat unusual to have so many genomes from so many different species. And we can start to ask, okay, well, what do the genes look like in these different species? Now, to do that, we have to do a couple things. We have to identify the genes in the species, right? So we have to figure out, okay, we know what they look like in C. elegans. Well, where are they? We have to search through the genomes using some of the tools, similar tools to what Dr. Norton mentioned. So what we did is we basically had these protein sequencing. We looked for proteins that looked like this. And let me say one more thing about this. So you notice how some of the letters are really big and some of the letters are smaller. Well, if the letter is really big, that means that these GATA factors need to have like C there and a C there. If the letters are sort of smaller, like you can see maybe here, G and D, that means that, well, you need to have a G or an N or a D there, or maybe a little bit other things, but really you have to have one of those, but it's not like it all has to be one of them. Like some of these GATA factors that combine GATA, some of them have a G, some of them have an N, some of them have a D. So we searched for, throughout all this genomes, we searched for proteins that look like this. And so we pulled them all out, and then we did something called alignment. So we have all these sequences, we have all these protein sequences, and then we try to figure out how to line them up. So we used various programs, and Dr. Norton talked about this when she was mentioning that, she took the patient's DNA and lined it against a reference genome. So that's sort of what we're doing here. We have all these different GATA factors, and we line them up, and you can see here's the C's that we were supposed to find right there, but there's some variation here. So we line them all up, and then we made what's called a phylogenetic tree. What is this? This is a tree of relatedness. So we try to figure out how each of these GATA factors, each of these sequences was related to each other. We basically grouped them. You can think like a normal tree, right? You have some branches that are very close together. You have some branches that are farther apart. Well, if they're close together, it means they're more closely related. If they're farther apart, they're more far distantly related. So here's what that looks like. Okay, so the central part of this is a phylogenetic tree. So there's sort of branches on the outside, these sort of, there are leaves on the outside. These are each of the sequences, and I basically have represented by a colored bar along the outside. And then you can see that there's some hierarchical relatedness here, like these are all more closely related to each other than they are to these, right? Or even than they are to these, but these two groups are more closely related than they are to, say, these groups over here. Okay, so we can, these are all these data factors we found. What I've done is I've color coded them according to their species, and the species is sort of broken up here. Here again, you can see this phylogenetic tree where these different species are more or less closely related to each other. So like the blue species are more closely related to than they are to the green. The blue and green are more closely related than they are to these things. So there's basically groups of different species that are related to each other. You might think about this like mammals, right? You have the primates, they're more closely related to each other than, say, the cats are, than they are to manatees, than they are to cetaceans. Okay, we have these different groups. Similar to what's going on here, although these species are much more like closely related than, say, we are to manatees. Okay. All right. So we have this phylogenetic tree, and these are all these data factors. Now, here's this network I talked about before. Okay. And what's sort of interesting about this is I've highlighted, so here's the network. These are the genes that are involved in intestinal differentiation. I've highlighted them on this tree. So here's one, here's the L7, that's here. Here's the meds, those are there. Here's N3, that's here. Here's L2, that's there. Here's N1 up here. Okay, and one thing you might note, if you look very carefully, is that there's a lot of blue and green, like N3 right here. It's all blue and green. And L7 and the meds, all blue and green. In fact, the only one of these that has any purple, red, orange, yellow, or darker purple is this L2 down here. So what that means is that none of these species down here have any of these core genes in this network. And this is the network that tells a gut cell whether it's going to be a gut cell. So you might ask, well, if they don't have the genes, how do they do it? If you've got to have a gut. So how do these worms make a gut if they only have one of the genes that tells them to do it? So that was our question. So let me show you what we did. So what we did is we looked at one of these species, and because we knew the sequence, we could use a technique that lets us actually fluorescently label molecules, fluorescently label RNA in individual cells in an organism. So here's a picture of that. So here I'm looking at this gene L2, and these little dots here are the expression level of L2 in individual embryos. And here are just how many nuclei there are. Basically, there's fewer nuclei here than there are here. These are late stage embryos, these are early stage embryos. And you can see that there is some expression of this gene in the gut, in the sort of center where the gut would be. So this is this late James, and this is one of these species that is sort of outside, which doesn't have the rest of the genes. Okay, so I'll skip that. So what it turns out is that these species down here, well, this L2 gene seems to work the same way. It's expressed in the right place. And we have some other data that shows it's doing the same thing. But what about all this stuff? Well, so the clue that we found by searching these genomes this way is that there's this gene L3 that's sort of it's closely related to these ones over here. In other words, these ones here, it's actually closely related to them, but in C. elegans, L3 is expressed in the skin cells. So it doesn't have anything to do with intestine. But we thought, okay, well, this L3 is in, so there's blue, green, and all the other colors too. So maybe in these species down here, maybe it has two roles, or maybe it's different. Maybe the evolution of this thing changed, or maybe this thing evolved. So we did is we looked to see where this is expressed in this species down here. And what we found is that there's actually two phases of this gene, L3. So here's what we saw before. And you see that early on, this L3 gene is expressed in the embryo, or in the intestines, or the middle. But then later on, it's expressed on the outside. not in the intestine like you see here. So later on, this gene's expressed in the outside. This picture, this late, is what it looks like in C. elegans. But early on, that's actually different from C. elegans. So this gene seems like it has this early phase of expression where it's expressed in the intestine, and a later phase of expression where it's expressed in the cuticle in the outer part. Okay, so basically from this analysis, we have this model where what happened was these species essentially have this L3 gene, and then they activate L2, and then they make a bunch of intestine. What happened was during the evolution of these species, this gene duplicated several times. So sometimes when you have you go through meiosis, and your DNA can sort of loop around and make a copy of itself. So you get, all of a sudden you have two genes that are identical. And then, but then they're there, and then they can start to, one can pick up mutations, they can start to diverge. And we think that happens several times here. So you had one gene that was multifunctional. It was involved in intestine and the skin, and then it duplicated, and they basically split those functions. So one of the copies stayed basically specialized in the skin. The other one duplicated many times to make all these intestine-specific genes. I didn't say one more thing about this, which is very interesting, which is all these worms, they look like they develop exactly the same way. Like if you looked at them by eye, you'd have a very hard time telling them apart, even if you were an expert. They look exactly the same. And so what's sort of neat is because we can go into the genome, because we can sequence them and use this technology, we can actually see that you can actually get tons of change at the genome level, a lot of change. I mean, you have like whole duplications of many, many genes. The network looks different. And yet, at the organism level, nothing seems to have changed. So there's this decoupling of the morphology and development from actually the molecules, which is neat to think that you can get changes in the genome that actually don't affect anything or don't appear to affect anything at the organism level. All right, so just want to thank Tony Daraga, the grad student in my lab, who did a lot of this work with some help from a high school student, Kevin Yem. And Louis Stevens and Mark Blackster were the ones who really drove the sequencing, who used the Illumina platform that Dr. Norton was talking about to really sequence these 60 genomes, and a great resource for the worm community. All right, that is the story of the worm.
Thank you, Dr. Rifkin. That was very interesting. I'm not gonna lie, I got a little bit lost because my background is I'm not a life science person, but it is really interesting, and it's really cool that we have, once again, this technology available to do all of these things. So yeah, it's really great. Thank you so much for sharing your presentation. So we do have questions. I know that Heidi had answered a few, so I want to just read through those just so everybody can hear them. And if you do have any questions for Dr. Rifkin's presentation, please do ask those in the Q&A function. One attendee asked whether the technique of sequencing techniques that Heidi spoke about in her presentation were cost-effective or accessible to hospitals. And I don't know how you want to do this, Heidi, whether you want to read your answer. Sure,
yeah, sorry, I realized after I was just so excited typing answers, I realized we should do it.
That is fine.
So yeah, so it is becoming more and more cost effective and accessible to hospitals. So you know, there's a few different options. There's whole genome sequencing, which is kind of what I talked about, where you're sequencing the entire genome. There's also exome sequencing, where you're sequencing only the region of the genome that codes for genes. So whole exome sequencing is used a lot more in hospitals and the clinic because you're sequencing a smaller amount. It's typically less expensive per sample. However, as the cost of sequencing goes down, Illumina is working towards a $100 genome. It becomes more and more cost effective to do whole genome sequencing that enables you to answer a lot of different questions instead of only sequencing a subset of what you might be interested in. And so there have been some recent studies that have found that hospitals who are able to implement whole genome sequencing actually end up saving money, even with the kind of startup costs of buying all this equipment. If they're able to do whole genome sequencing for their patients, they can get a faster diagnosis, and it ends up kind of reducing healthcare costs. So we're definitely moving in that direction, but I would say that doing whole genome sequencing in a hospital setting is still at the very early stages, but I think we'll see a lot more of that pick up in the next 10 years.
Thank you. And then another question was, do the computer programs that you use to analyze the genome also end up determining what the genetic disease is, or, is there more work after that stage?
Yeah, that's a great question. So that final step of kind of interpretation that we talked about, where you're using these knowledge bases and there's software programs that can do that for you. So you can plug in your VCF, your variant call file, and then it can look at the different databases that you tell it you want to look at and you give some information about the sample, like maybe this is a cancer sample or a genetic disease that you're testing for, but ultimately it's not going to spit out a diagnosis. It can just give you some information about the variants. And really that's where this final stage is, where you have to have a lot of expertise of either a geneticist or a clinician to kind of interpret those results. And so it will give the results in an output that's much easier for a human to digest and understand than that speckled file that we saw. But ultimately there is some human steps needed to kind of make a decision. You have to kind of integrate the information you know about the patient and the data that you're seeing about these types of variants.
Great. And I think this one might be directed towards your presentation, Dr. Rifkin, but an attendee asked, are the mutations that you spoke about essentially silent mutations because they do not affect the phenotype? Because I know you mentioned at the end of the presentation how the worms look the same despite the genetic difference.
Yeah, so it's a good question. So yes, so they don't seem to differ morphologically. Where it's not so clear is whether this more elaborate network, it might be more resistant to new mutations. For example, if I just have like one gene activating another, and I get a mutation in any one of those, I could destroy the whole thing. But if I have sort of multiple routes to activate the downstream gene, it might be more robust. It might be more less sensitive to mutations. And so it sort of just depends on how exactly you want to define your phenotype. But it's an interesting question that it doesn't seem to have an effect, but it might make it like it might lower the risk, one might say. Yeah.
Thank you. So yeah, that was answered. Okay, so I guess this would go to both of you. Are there any risks or cons to genomic mapping? I know we talked about, or Heidi, you mentioned the pro being the cost-effective measures, and Dr. Rifkin, you talked about, allowing us to identify things further, aside from the phenotype. So can you all think of any risk or cons? Or is that a little controversial?
No, I think it's a good question. So, I mean, really, the risk is that you might find things that you don't know what to do with, right? So you find mutations that maybe are related to some genes that you see have something to do with Alzheimer's disease, for example, and you don't really know what that actually means. So if any of you have kind of heard of any like 23andMe or some consumer genomics type companies where you can send a sample in and they'll tell you genetic information about yourself, they actually carefully control what information they'll give you versus not, because they can only give you information about things if they actually know what to do with that. So if you were to sequence your genome and get the list of all those variants back, you might run yourself into a very concerned state as you're kind of looking up, what do all these different mutations mean? Will this mean I'll get cancer? Will this mean I'll get Alzheimer's disease? And so really being able to make sense of that data And that's where that really highly trained geneticist, genetic counselor can help make sense of that data. So that's the risk, is uncovering information that you don't know what to do with, I would say.
I'll jump in one more thing. So one of the really neat applications of this technology that's been developed the last 10 years, I think, is where people have gone and they've actually started sequencing like really, really, really old samples. So, you get a sample from somebody who lived 1,000 years ago or 2,000 years ago, not just people, but organisms too. And there's this whole field of paleogenomics where you're trying to extract DNA and figure out, okay, what were the genetic variations a long time ago? Like what sort of diseases were there? And also people have been using this to track how humans have migrated around the world. Because you can actually use all this data to really figure out human migration patterns. Dr. Norton mentioned this 23andMe, and another thing that these companies give you is your ancestry report, right? And certainly you can look up on YouTube, if you want to know cons, you can look up on YouTube and watch people opening these things and discovering they're not who they thought they were. Well, you can ask, okay, what does that mean they're not who they thought they were? But certainly for some people, when they get these reports, it has a big impact on them. And you can debate whether they should or not, but it does. And so that could be a con. But I think that there's a lot of interesting science out there about how to use this to really track the sort of recent history, recent being, the last 20,000 years. I'm an evolutionary biologist, that's recent. The recent history of humans and thinking about that. So that's sort of a neat application that's becoming more and more possible as the technology has really been developed and is more and more powerful.
Really great answers. The next question is, what programming languages are the algorithms typically made of?
That's a great question. So I would say Python is a very common, R are very common languages to use if you want to kind of write your own algorithms. But some of the, to make things a lot more efficient, there's, C is often used, and actually the type of software that I work with, actually, this is probably too much of a deep dive, but it actually runs on a special type of hardware called field programmable gate arrays. And so it uses C language, but then also like hardware descriptive language. So it's really kind of like electrical engineering type work. But I would say if you're interested in getting into this type of data analysis and want to understand some of these software programs, Python or R would probably be the best languages. I don't know, Dr. Rifkin, if you have other recommendations of other languages.
Yeah, definitely. Exactly what Dr. Norton said. If you want to learn how to do this, learn Python or learn R. The great thing about them is they're free, and there's like tons of people writing stuff for them, right? There's lots of information on the web. So they're relatively, they're designed, the way you write, they're a lot easier than C because you don't have to deal with a lot of finicky stuff. So they're a great way to learn how to program if you don't know how to program, and they're also really powerful, right? This is what people use in general for do a ton of things. And then of course, as Dr. Norton said, there are specialized applications where you might want to make something run faster, you have to particular architecture. But definitely, if you want to learn how to program, learn Python or R, especially for bioinformatics and biology.
That's that interdisciplinary nature of STEM. Look at that. It all goes together. Okay, so our next question is it possible to use your technology during the embryo, I guess, phase, so we can use preventative care, or is it only possible after birth? As in, if you detect a mutation, would it be possible to then use CRISPR to fix it?
So the idea of testing the embryo is possible. So that's actually done today for kind of in vitro fertilization. If the parents have a known mutation and they know that that their offspring has a high risk of having a really serious disease, they can do screening of embryos so that they can kind of select which embryo they want to implant or transfer. So that's a common already used today in the clinic to do that type of screening. The next step of detecting if there's mutations to use CRISPR to fix it, there's a lot of ethical considerations there. There have been reports that has happened, but it's been kind of outside of the scientific community norms. I think that in general, most people think that CRISPR is not ready for something like that, and there's a lot of ethical considerations to think through before we do something like that. But conceptually, that's kind of, you're linking together all the right concepts of what could be chained together.
Yeah, so we use CRISPR on worms in my lab, and one thing you have to be careful with the technology as it is right now is that you want to make the specific change, but oftentimes, not always, but oftentimes you make other changes too, right? And so if you're doing that in humans, right, you may fix the thing you want and you may create other problems. So CRISPR technology is awesome, but it's probably not it's probably not precise enough quite as it is right now to use it in humans.
Great answers. And I think that ties into the next question really well. An attendee asked, can the processes or techniques you use to study the worms also be applied to humans in the future?
Well, certainly the genomic processes can. And in fact, people are sequencing tons and tons and tons of humans. And that's sort of at the base of what Dr. Norton mentioned, right? You find all these variants, right? And how do you find like a rare disease variant? You have to have lots and lots of sequence. You have to know what's sort of normal, what's not, and then you have to know which variants are linked to particular diseases, right? So you need to sequence a lot of people in order to make those associations. So that's one way where, it's the same in whatever organism you're studying. The sort of the visualization techniques that I use. So we're mapping, or we're sort of looking where particular genes are expressed. So people do use that. I mean, not so much not in like human embryos. I don't think that would be ethical, but in human stem and sort of cell culture, people definitely want to see what's being expressed. And there's a lot of neat technologies in human cell culture where you can actually create 3D cells that are sort of developing into some tissue. There's a lot of advances in that. And you want to see, okay, what genes are expressed. Like if I want to make a kidney, what genes tell the body it should make a kidney right there, for example. So that's there too. And the phylogenetics thing, yeah, so that also is used to analyze human evolution.
Thank you. I think that there are two different questions that kind of relate to each other, Dr. Rifkin. One of the questions is, why do we use nematodes to study developmental biology? And the follow-up question is, are GATA factors present in the human genome or just in nematodes?
Okay, yeah, so GATA factors are actually present like throughout organisms. So humans, for example, have 3 GATA factors that are involved in intestinal development, in blood development, and all sorts of different things. It's just, for some reason, these particular nematodes have lots of them. And so we're not quite sure why they have lots of them. So yes, so they're a common type of transcription factor, a common type of one of these regulatory proteins. Why use nematodes to study developmental biology? So one nice thing about them is that we know what every cell is. There's only 1,000 cells in these worms, and we know what every cell is supposed to be. So it's very easy to figure out if something goes wrong. What that means is that if we can mutate a gene and we see that something goes wrong, it's sort of easy to make that connection. So that's one really nice thing about using these nematodes, is that development, and they're also very small. We can watch them develop under the microscope. They're transparent. So there's a lot of nice things that make them very convenient. So their genome, they're transparent, they're easy to grow in the lab. It's easy to tell if something goes wrong in development. So all those reasons, nematodes are really nice. These particular nematodes are nice for developmental biology.
And I think we have one last question. If various genetic patterns we see not impacting the organism, how do we prove that a specific genetic mutation causes a specific disease? Are these outliers that are these, I guess, how do we recognize these outliers?
So I guess
that's what you talked about, Heidi, like the specialization after that stage, but I'll allow you to elaborate.
Yeah, so really it comes into how do we build out these databases that allow us to assess the impact of a particular mutation. That's kind of how I interpret this question. And so it's basically the type of work that Dr. Rifkin's doing of understanding how different genes are working. You can do it in a developmental organism. Then you can also study genomics across a population where you get a large sample size. You can see how maybe if you sample, you look at 1,000 different patients, you sequence their genome, and you see that all of them that have this particular mutation have trouble metabolizing a certain drug, for example, then you can start to build evidence that this mutation and this phenotype are linked. And so that's, part of the scientific, the goals as we work to understand the genome and the impact that it has is we're constantly building up our knowledge that can feed into these types of databases to allow us to interpret the significance of some of the variants that we're detecting. But it has to start with some type of experimental, an experiment, a study that's kind of asking a question and uncovering some biology.
Right. That was all of our Q&A or questions. That's the Q&A session, unless you all have any other questions. But let me share my screen.
There was one I answered in the chat because I was worried that we were going to cut it off and not get a chance to. And so I thought this was a very insightful question about making sense to have multiple reference genomes so that you can have a more personalized reference for the patient. That's a very insightful question, and that's an active area of work. There's a lot of different ways that you can do this. One of the ways that we're working on it at Illumina is working on kind of a reference genome that incorporates information across a lot of different individuals that come from a lot of different ancestries, and perhaps even tailoring, what part of the, which information feeding into the reference genome you use based off of the patient that you're working with. So that's a very insightful question, very active area of work.
Thank you, Heidi. And yes, thank you everybody else for joining us, all attendees. Once again, thank you to our panelists, Dr. Rifkin and Heidi Norton. We appreciate, all of the information that you all just shared with us, that you both just shared with us. I learned a lot today, and I'm sure that our attendees, based off the questions, it was very informative to the attendees as well. This event has been recorded, so as it says in the chat, we will send notice when the recording is available in case you want to go back and re-listen to this or re-listen to some of the questions that were asked, just to get a better understanding of this this technology that's used. But yes, go to our website, the CE website, so you can register for our upcoming events. And once again, thank you to Illumina for helping us put together this this event tonight as well. Yes, thank you everybody so much, our attendees, and once again, our panelists. I hope you all have a great rest of your day.